Welcome. In this session, I'm going to discuss about DNA replication. So let's begin this session with DNA replication. Now, what is the DNA replication? So let's first look at the definition here. So it is a process of making an identical copy of a DNA from parental DNA. Is a process of making an identical copy of a DNA from parental DNA. And the mechanism for this DNA replication is actually proposed by J.D. Watson and F.H.C. Crick, who proposed the double helix model also. So the mechanism was given by Watson and Crick. So they have given the mechanism for this DNA replication. And what they told is that DNA replicates by semi-conservative method. So it replicates by semi-conservative method. I'll come to this point shortly. So before that, I would like to remind you, in the last session I told that DNA replicates by itself. So it has a property of making its own copy. So that's what the DNA replication. And this DNA can also make RNA molecules in a process called transcription. And this RNA will undergo translation that results in finally the formation of a protein, right? So this is what called central dogma of molecular biology, and in this central dogma of molecular biology, the first part, the DNA replication, that is what I'm going to discuss now, and after that we will see the transcription, and after that we will also see about translation process, right? Now coming back, Watson and Crick, I told you, they suggested that DNA replicates by semi-conservative method. So what we mean by this semi-conservative method, so let us understand it first. So here I'm going to take a parental DNA, so with some bases I'll just take, right? We know that the two strands are complementary to each other, G, C, okay. So this is 5 dash to 3 dash, this is 3 dash to, okay, 5 dash. Right, now this is parental DNA. Now during the process of replication, what happens is these two strands, they get separate. So that's what I will draw it separately. The two strands will get separated. So A, C, G, C, T. So 5 dash to 3 dash. Okay, this strand got separated. And again, this is another strand. Okay, T, G, C, G, A. Now, right, this is the one that acts as a template strand. So this is a template strand, and with the help of this template strand, a complementary strand is going to get synthesized. So that will be a new strand. So a new strand will be synthesized. So we know that if we know the sequences on one strand, we can predict the sequences on opposite strand. So by using this strand, a new strand will get synthesized, right? So this will be 3 dash to 5 dash. Okay, clear? And this also, in this also the same, here A, so this is C, G, C, A. Right, now finally it resulted, okay, from one DNA, okay, from one parental DNA, two daughter DNA molecules are formed. So two daughter DNA molecules are formed. So there are two DNA daughter molecules, and in this, the okay, one is the template strand. So this template strand is actually the parental strand, or I'll say the old strand, and this one is the new strand. So this is the new strand, so newly synthesized strand. Okay, so whenever a new DNA molecule is formed, that will be having one old parental strand and one newly synthesized strand. So as every DNA molecule, even this parental DNA also has the same. Okay, so here I did not show it, okay, just to make you to understand that, and every time a new DNA molecule is formed, one will be the parental strand and one will be newly synthesized strand. So hence, right, the DNA replication is called the semi-conservative. So it's called, right, semi-conservative method of DNA replication. Now question comes, what is the proof that DNA replicates by semi-conservative method? So that is what this was experimentally proven by two scientists, Matthew Messelson and Franklin Stahl. So that's I call Messelson and Stahl experiment. So now let's see the experiment and find out how they proved that DNA replicates by semi-conservative method. So let's see Messelson and Stahl experiment. Messelson and Stahl experiment. So this experiment proved that DNA replicates by semi-conservative method. Replicates by semi-conservative method. And this is proposed by two persons. So that is Matthew Messelson and Franklin Stahl. So these are the two persons who proposed this experiment, and that was in year 1958. In 1958. So let's see what they have done, okay, and how they proved that DNA replicates by semi, okay, conservative method. So they grew E. coli on a medium containing N15H4Cl. So this is called the ammonium chloride, and it has heavy nitrogen. So that is isotope. So this N15 is heavy isotope of nitrogen. So N15 is a heavy isotope of nitrogen. Okay, and what they did, they have grown this E. coli on this on a medium that has N15H4Cl for several generations. So let's see here. Okay, so in a petri dish, so this is the nutrient medium, and that contains, okay, N15H4Cl. So it has N15H4Cl for several generations. So these are all bacteria growing on this, okay, nutrient medium. So E. coli bacterial colony. It's called E. coli bacterial colony. And I told you that they grew E. coli on a medium containing this for several generations. For several generations they have done this. Okay, and what happened, after several generations they have taken this E. coli from this N15 medium, and then they extracted the DNA from E. coli that were grown on this N15H4Cl. Okay, and so let's see here, this I'm taking is a DNA molecule. Okay, and in this one strand is having N15, the other strand also has N15. So what happens when replication takes place, right? Whenever a new strand of the DNA is forming, okay, it uses the sources. So here in the source there is availability of N15, and we know this N15 will become the part of nitrogen base. Okay, so this N15 will become part of nitrogen base. So every time there will be synthesis of nucleotides. We know a nucleotide is a combination of phosphate, then the pentose sugar, and a nitrogen base. All these three, okay, have to be synthesized every time whenever there is a DNA replication process. Okay, and during that synthesis, it will use this N15 to make these nitrogen bases, part of nitrogen bases. And after several generations, when we extracted the DNA from the E. coli that were grown on this nutrient medium, they're having N15H4Cl, okay, so both the strands will be having N15. So how we have come to know it, so we did a density gradient centrifugation using CSCl. So that is cesium chloride. So I have taken in this test tube CSCl. Okay, so CSCl stands for cesium chloride. Stands for, okay, cesium chloride. And now I did density gradient centrifugation. Now when I did density gradient centrifugation, what I observed is that once I finished this, I got a band, okay, near to the bottom of the test tube. So I got a band near to the bottom of the test tube, and because as N15 is heavy and both strands are having heavy isotopes, it came near to the bottom of the test tube. So that's why I will mention here N15, N15. So why two times? Because indicating the two strands. Okay, so this is what we have seen for the E. coli that were grown in the nutrient medium having N15H4Cl for several generations. And one more point is that E. coli divides for every 20 minutes. E. coli divides for every 20 minutes. So now what I'm going to do is the E. coli bacteria that are grown on N15H4Cl, okay, so they are shifted to medium containing, N14H4Cl. Okay, so this one has N14H4Cl. So in this case, the N14 is lighter isotope of nitrogen. So it's a lighter isotope of nitrogen. So what I did, I shifted or transferred the E. coli bacteria from this N15H4Cl. I means here the scientists, okay. So they shifted here N15H4Cl from this medium to this lighter isotope, that is N14. So here I mean okay, so shifted here. So now these are all E. coli, and wait for 20 minutes after transferring. Okay, so they waited for 20 minutes and then they took this E. coli, right, and again extracted the DNA, and then they did, okay, density gradient centrifugation. So now let us see how they obtained this density gradient centrifugation. Right, so this one, after 20 minutes I'm going to get a DNA that will be hybrid. Okay, so this is the DNA molecule that is obtained, and you can see in this case this is having lighter nitrogen, okay, and in this case this is having, okay, heavier one. Now doing density gradient centrifugation, so when they did density gradient centrifugation, what they observed is that they obtain a band at the center of the test tube. So this is CSCL, and here they obtained a band. So this one will be, right, N14 and N15. N14 and N15. So when they took, okay, the DNA from the E. coli that were grown on N15, they obtained a band near to the bottom of the test tube, but in this case they obtained, okay, right at the center. Now they continued for 20 more minutes. Okay, so they allowed one more generation. So right, so this will be the first generation of E. coli bacteria, and you are going to get a hybrid because it has one strand with N15 and another strand with the N14. Okay, so that will be the first generation, or I'll say this is generation 1. Now they continued the E. coli to grow on this medium for 20 more minutes. Then let's see. So that is okay, 20 more minutes. So here 20 minutes, this I'm going to take 40 minutes. Okay, right. Now after the 40 minutes, what I have seen, okay, so this strand is going to become, of course, into right 2. Okay, and in this case a new strand is going to get synthesized. So here one already parental strand and one newly synthesized strand. So this is how you obtain. So this will be N15 and this is N14. This is also N14. This is also N14. Okay. Now when they did again density gradient centrifugation, in cesium chloride, CsCl, and what they saw is that they obtained, okay, right, one band near to the top of the test tube. So this is one band and one band that was already seen at the center. Similarly, like we have seen in the previous experiment, they obtained. So this is actually N14 and N15 molecules, that is DNA hybrid molecules are seen in that case, and this will be N14 and N14. So it means that in this case this is lighter DNA molecule, whereas this is hybrid DNA molecule. But when we see this one, this is heavier DNA molecule. So this is a heavier one, this is the hybrid, and this is the lighter one. So by obtaining, okay, two bands, okay, on the top, okay, near to the top of the test tube and one at the center, you can see if the DNA strand is completely formed new, then you will see either this strand, this band or this band. You will not see this band at all. And with this, Matthew Messelson and Franklin Stahl, they proved that DNA replicates by semi-conservative method. Right? I hope it's clear. Now, similar experiments were done by Taylor and colleagues. So you see, similar experiment was done by, okay, Taylor et al. So there are, okay, his colleagues, all of them together, they done an experiment on Visia faba, on a plant called Visia faba. Of course, in short, we can call faba beans, right? And they done this experiment by using radioactive thymidine. So in this case, they have taken radioactive thymidine, and they observed that one strand, that is newly synthesized strand, is only having the radioactive thymidine. And with that, Taylor et al. Also proved that the DNA replicates by semi-conservative method. Okay, so this experiment also proves that DNA replicates by semi-conservative method. Replicates by semi-conservative method. Okay, so that's all. Okay, the proof that DNA replicates by semi-conservative method, and we know that whenever a DNA molecule is formed, that will be having one old parental strand and one newly synthesized strand. Okay, so now let us move on to the actual process of replication. So how this DNA replicates and forms its own copy, that I'm going to discuss the DNA replication in E. coli. Okay, so prokaryotes have DNA replication a little bit complicated. Okay, first we need to understand how simply the DNA replication takes place. That we will understand by studying prokaryotes, and we know E. coli is a prokaryotic organism. So first we will understand in these prokaryotes how the process of DNA replication takes place. This DNA replication in E. coli, it requires several enzymes. So it requires several enzymes. I'm going to mention few of those enzymes what it requires, like gyrases, that are also called topoisomerases. Okay, so these are enzymes. Then it also requires helicase enzyme, then an enzyme called DNA dependent DNA polymerase. It's called DNA dependent DNA polymerase. So this I'll just mention as DNA polymerase 3 is the enzyme that is required. Okay, this is actually the main enzyme which is very important. Then DNA ligase, and lastly enzyme called primase. So these are different enzymes which are required in the replication of DNA in E. coli. So let's see what are all the functions of these enzymes. The first one, the gyrases or topoisomerases are the enzymes that actually relieve tension during uncoiling of DNA. We know that DNA is coiled, and that's what it forms the helix. Okay, so double helix, we know in that it will be seen in this way. Now for the replication to take place, this should unwind and it should become something like this. This one should get separate and this strand should get separate, right? The two strands should get separated when the DNA replication takes place. It is not necessary that first the entire length of the DNA will get separated. No, that I'll discuss shortly. Okay, you can see this is a part where the DNA uncoiled. So when this DNA uncoiling, it can produce tensions on both sides. To relieve that tension, these gyrases are used. So topoisomerases will help in relieving tension during uncoiling of DNA. Then coming to helicase enzymes, so they separate two strands and break hydrogen bonds. We know these hydrogen bonds in DNA, they exist between nitrogen bases. So the function of this helicase is to separate the two strands and break the hydrogen bonds. And coming to DNA polymerase, so this is the DNA replication enzyme. It's DNA replication enzyme. So that's what it adds nucleotides on newly or so it adds nucleotides on the new strand. Of course, by adding the nucleotides it forms the new strand. And coming to the DNA ligase, this one helps in joining fragments of DNA. So that one helps in joining fragments of DNA. And lastly, the primase enzyme. So this one helps in formation of RNA primer. So it helps in formation of RNA primer. So that RNA primer will be a short stretch of nucleotides, and it provides, the RNA primer actually. So this RNA primer provides third OH ends to DNA polymerase 3 enzyme. So it will provide third OH ends to DNA polymerase 3 enzyme. So that's the functions, mainly the enzymes which I'm going to discuss in the replication of DNA in E. coli bacteria. So these are the enzymes, and I have mentioned their functions. Now I want to add a few more points on this DNA dependent DNA polymerase that I call DNA polymerase 3 enzyme. So here there are some specialities with this enzyme, that is DNA polymerase 3 enzyme. So what is that? DNA polymerase 3 cannot start, it cannot start a new strand by its own. The DNA polymerase 3 enzyme doesn't have the ability to start a new strand. So it cannot start a new strand by its own. Say suppose we have one DNA strand here. So the new strand has to start synthesizing, and this is not, or it cannot be initiated by DNA polymerase 3. So someone have to initiate, and that someone is this RNA primer. So this RNA primer will be formed with the help of enzyme called RNA primase. Will result in the formation of a short stretch. So this is, say suppose, the RNA primer, and that will have 3 dash, and this third end is where the OH is available, and this DNA polymerase 3 can add a new nucleotide only on third OH end. So that is what one more point I say. It adds a new nucleotide only on third OH end of a pentose sugar. So it requires that third OH end. So if that third OH end is not present, it is not going to add a nucleotide. right? So this is what all the enzymes and their functions. And most important enzyme is this DNA dependent DNA polymerase. This is the main enzyme that actually, okay, helps in synthesis of new strand. Of course, there are certain issues with this DNA dependent DNA polymerase. What is that? It cannot start a new strand by its own, and it requires third OH end. So without that, it cannot uh, right, perform synthesis of a new strand. Right. So let's see, okay, how this takes place. Now, the replication begins at a point or at certain point. So replication begins at certain point. So it is not like the replication will begin at anywhere on the long DNA molecule. It is not the case. There will be a specific location where the process of replication begins on the DNA molecule. So that, okay, special location where the replication begins is called the origin of replication. So in short, it's called ori site. The origin of replication, in short, called ori site, right? So let's see. Say suppose at that point the ori site, okay, so that will be having some special sequences that can be identified by helicase enzymes. Okay, so this will be with some special sequences of nitrogen bases. So that will be identified by the helicase enzyme. And say suppose that helicase enzyme is bound at that point. Actually, uh, these two, okay, are like this. And say here the helicase enzyme bound, and it started separating the two strands and also started to break the hydrogen bonds, and it is moving in this direction. And because of this, so the two strands got separated, and say this is where the helicase enzyme is present, and this is the direction, okay, how it is moving. So in this direction it is moving, and from, okay, from this point, say suppose this is the origin of replication, thereby, okay, separating the two strands, creating a replication fork. Okay, so this is what it has created, a replication fork. Okay, so now let's take the polarity of the strands. This is important, very very important. Without this we cannot explain the process of replication. Okay, so now, okay, so this is the strand, okay, that has a polarity of 5 dash to 3 dash, and this one has 3 dash to 5 dash. Now let's see, okay, on this strand how the replication begins. I told you that the problem with the DNA polymerase 3, so DNA polymerase 3, that it cannot start a new strand by its own, hence a primer need to be synthesized. Say suppose this is the end here. If I'm going to if the primer is going to form here, so this is this will be the 5 dash, and this one will be the 3 dash. So it means that this is point where OH end is available, and this is a primer. And now when this is available, right, the third OH end, the DNA polymerase 3 will bind here, and it starts adding nucleotide by nucleotide. So you have to keep in mind a new strand, okay, length increases by addition of nucleotide by nucleotide. One by one nucleotide will get added. Okay, so that's what we know the nucleotide, how it looks like. So this is a nucleotide, and we'll have phosphate. So phosphate and phosphate. So this is sugar, and this is you can take either adenine or guanine because there are two rings. Okay, if there is single ring, then you need to take cytosine or thymine. Okay, now these are called deoxyribonucleoside triphosphates. So they are called deoxyribonucleoside triphosphates. We know that this part, that is the pentose sugar, so this part pentose sugar, and the nitrogen base, this part will be the deoxyribonucleoside. And these are three phosphates, the triphosphates. Okay, and the bonds present between these phosphates, the last two phosphates, so these are high energy bonds. High energy bonds. So they contain energy, and of course when this is going to add up here, okay, these two terminal phosphates will get removed, and this will become monophosphate. So the deoxyribonucleoside triphosphate will become deoxyribonucleoside monophosphate. Remember when I told you the nucleotides of a DNA molecule. Okay, the deoxyribonucleoside triphosphate will become the monophosphate. And these deoxyribonucleoside triphosphates have dual functions. Okay, so what is that dual function of this deoxyribonucleoside triphosphate? One, it acts as substrate. So this is what it's going to get added. Of course, the last two terminal phosphates will be eliminated. So, and when they break up, the hydrogen bonds, the bonds which are present between these two phosphates, when they break up, and they will release energy. Okay, of course they are not hydrogen bonds here. Okay, they release energy, and this monophosphate is going to attach here. So that's why I said it acts as substrate, and they will be also acting as source of energy. So the second one, so they are source of energy for polymerization. Source of energy for polymerization. Polymerization means nucleotide by nucleotide addition that results in increase in the length of the strand. That is what we call polymerization. So this deoxyribonucleoside triphosphate will act as substrate and also act as a source of energy for polymerization. Now this is DNA pole 3 enzyme, and you can see here one nucleotide is added. Then again this nucleotide will have 3 OH end. Then again it adds one more. Then adds one more, adds one more, and you can see in this case it goes on adding nucleotide by nucleotide, and this is how the strand grows, okay, on one of the strand. Then coming to the opposite strand, so in this case opposite strand, say suppose if I'm going to make a primer here, then this side I will be having, okay, 3 dash, and this side it will be 5 dash, and say on this side this is the end, say suppose, okay, I cannot add the third OH, I mean the DNA polymerase 3 will not be able to add further because there is no strand here, okay, say suppose. Then now on fifth end, so this is the fifth end available. Now this fifth end is of no use because I told you the DNA polymerase 3 will never add a nucleotide on 5 dash. It will add the nucleotide only on the third, okay, or 3 dash, that is third OH end. So hence this primer, say, is formed here, okay, so this is 5 dash and now 3 dash. Now the DNA polymerase 3 binds up here, okay, so this is the DNA polymerase 3, and it starts adding nucleotide by nucleotide, and this is how okay, so this is the 3 OH and you get clear. Now say this helicase of course will be going on and this 3 OH end will be always available, so hence the DNA polymerase 3 actually it is not present here. By the time it comes up to here, it will be on the top here and goes on adding and adding, and say this has almost come up to, okay, this point. So here is the DNA polymerase 3 enzyme. Okay. Now, and let's mention here DNA pol 3 enzyme. Now I told you this side is of no use. Hence, what need to be here? Again, one more primer need to form. This will have again 5 dash and 3 dash. Now the DNA polymerase 3 binds up here and then starts adding nucleotide by nucleotide, and it will be growing up to here, right? So for this strand only one primer is enough, okay, but on the opposite strand one primer is not enough. Now say this helicase, now it has moved up to here, then it will expose, right, some more some part of the strand again. Now here already 3 OH end is available, there is no problem, this will continue, so it will go on, okay. So here I'm not separating it, actually it will separate, okay, as I need to disturb all this, that's why I'm not drawing again. So it will continue on this, but again you can see in this you will get 5 dash, there is no use, hence again one more primer has to form here. This will have polarity 5 dash 3 dash. Now the DNA polymerase 3 binds up here and then starts adding and adding nucleotides one by one, not all at a time, one nucleotide by one nucleotide. So for this reason, this strand will be going on, okay, synthesized very quickly. So as there is only one primer required, okay, there won't be any problem in this strand. Hence, the synthesis of this strand will be faster when compared to this. Hence, okay, the strand on which there is continuous synthesis, so on this there is continuous synthesis, it's called leading strand. So this is called the leading strand. Okay, and on other case where on this strand the new strands are synthesized in fragments, so hence this is called discontinuous synthesis. And as it is slower when compared with this, it's called lagging strand. And on this lagging strand, the fragments of this DNA are actually discovered by a scientist named Okazaki, and hence, so these fragments are called Okazaki fragments, discovered by scientist named Okazaki. So he came to know that on one strand, the newly synthesizing strands will be synthesized in fragments, not at a stretch. Only on one strand, the newly synthesizing strand will be at the stretch, whereas on opposite it will not be. Right? So that's why this becomes the discontinuous strand. You can see on discontinuous strand many RNA primers are required, and if this helicose goes somewhere up, and again one more RNA primer is required, and from that point one more Okazaki fragment will be going on synthesized. And once it comes to the end, now one DNA molecule you can see easily becoming two DNA molecules. And this entire process of replication, it completes in 18 minutes. So entire process of replication, it completes in 18 minutes in case of E. coli. It means that in this case there are 2000 base pairs formed per second. So 2000 base pairs are formed per second. So approximately this is approximately 2000 base pairs are formed per second. The entire process of replication in E. coli completes in 18 minutes. So this is all about the DNA replication in E. coli. Right, so with this I will end this session. In the next session we'll see the next part of the central dogma of molecular biology, that will be transcription.